In 1976, NASA placed a tiny laboratory on Mars, fed nutrients to the soil, and watched it react as if something were alive. This is the 50-year-old cold case of the Viking 1 lander. One experiment seemed to scream, life. But another, more trusted instrument reported the soil was completely sterile, containing no organic molecules at all. NASA faced a contradiction. And with July 20th, 2026, marking the 50th anniversary of that landing, the case isn't just cold, it's deep frozen. So today, we're reopening the files to ask, what did Viking really find? And why is this 50-year-old result more controversial now than ever? To understand the mystery, you have to appreciate the challenge. How do you even begin to look for life on a planet where you don't know what it looks like, how it behaves, or what it's made of? You can't just send a microscope and hope to see little green men waving back. So NASA's strategy was brilliant. Instead of one big all-or-nothing test, they packed several smaller, different experiments, each looking for a different sign of life, multiple shots on goal. One of these, the labeled release experiment, was brilliantly simple. It was like giving the soil a bowl of soup, a liquid nutrient broth, full of simple organic molecules that Earth microbes love to eat. But here was the clever part. The carbon in those nutrients was tagged. It was radioactive carbon-14. This acts as a tracer, a way to follow the food through any potential life process. The theory was simple. If anything in the soil was alive, it would metabolize the nutrients and release waste gas, just like we do. Because the food was radioactive, the gas would be too. A detector was poised to spot that radioactive gas, and if it did, an alarm would go off. And on July 28, 1976, 8 days after landing, that's exactly what happened. The alarm went off. A sharp, immediate spike in radioactive gas, released from the Martian soil just hours after it was fed. On Earth, the science team was stunned. The curve of the gas release looked textbook. It wasn't a sudden chemical fizz. It was a steady, sustained release that gradually leveled off, exactly as you'd expect from a population of microbes consuming a limited food source. Gilbert Levin, the experiment's designer and principal investigator, was ecstatic. He had spent years developing this experiment, testing it in the harshest environments on Earth. He knew what a biological signal looked like, and for him, this was it. But science demands skepticism. To rule out simple, non-living chemistry, they had a brilliant control test built into the plan. This is what would separate a chemical fluke from a biological fact. The lander's robotic arm took a fresh sample of Martian soil, but this time, before adding the nutrients, it baked it at 160 degrees Celsius for 3 hours, hot enough to kill any known microbes. After it cooled, they repeated the experiment exactly. This time, nothing. The gas release flatlined. The reaction was gone. It was a textbook biological result. Activity in the fresh sample, no activity in the sterilized sample. This was the clincher. It met 2 key criteria for life. It showed signs of metabolism, and that activity could be stopped by sterilization. For a moment, it seemed undeniable. Back on Earth, Gilbert Levin was certain. He famously declared that the criteria for detecting microbial life on Mars had been met. It was a historic, world-changing moment. Except, there was one huge problem. Remember those other shots on goal? Another experiment on the lander was about to report its findings. This was the Gas Chromatograph Mass Spectrometer, or GCMS. And it wasn't looking for a term reaction, it was looking for the body. Think of it as a crime scene investigator for molecules. Its job was to identify the specific chemical compounds in the soil, to find the molecular fingerprints of the building blocks of life. Specifically, organic molecules. Carbon-based compounds that make up every living thing we've ever seen. If there were microbes in the soil, there should also be the molecules from their dead relatives. The evidence of their existence. The GCMS scooped up some soil, heated it to release its chemicals, and analyzed the results. And it found nothing, no organic molecules, at all. The soil was, by this measure, completely and utterly sterile. This was the contradiction. A scientific standoff. The labeled release experiment was screaming, metabolism. The GCMS was screaming, no bodies. Both results couldn't be right. Or could they? In the end, NASA and the broader scientific community sided with the GCMS. The logic was brutal and simple. No organic molecules, no life. Life as we know it is carbon-based. Finding a biological process without the biological building blocks seemed impossible. The conclusion was that the labeled release result must have been caused by some kind of exotic, hyper-reactive chemistry in the Martian soil itself, something that mimicked life when it got wet and warm, but wasn't actually alive. And just like that, the most exciting result in space exploration history was dismissed. Gilbert Levin became a lone voice, a dissenter. For the rest of his life, He would argue that his experiment had found life, but the world had moved on. The Viking life detection mystery was officially solved, closed. The answer was no, and for almost 30 years, that's where the story ended. The case was deep frozen. Until 2008. That year, a new robot landed on Mars, the Phoenix lander. Its mission wasn't to look for life, but to analyze the soil and ice in the Martian Arctic with a new generation of chemical instruments. And in that soil, Phoenix found something that Viking had completely missed. A strange and abundant salt called perchlorate. On its own, at cold Martian temperatures, perchlorate isn't that exciting. But when you heat them up, they become powerful oxidants. They aggressively break down organic matter. Suddenly, a horrifying possibility clicked into place for scientists. And how did the Viking GCMS search for organics? By heating the soil to over 350 degrees Celsius. The no organics result from 1976 suddenly had a new, deeply ironic explanation. If there were both organics and perchlorates in the soil, the GCMS would have incinerated them on contact. Viking may have been systematically burning the very evidence it was sent to find. And this isn't just a theory. Experiments on Earth have confirmed it. When scientists mix small amounts of organics with perchlorate-rich soil and heat it in a Viking-like instrument, the organics vanish. They get the exact same, nothing found result. This discovery changed everything. The Viking cold case was suddenly wide open. The main piece of evidence against life was now seriously, perhaps fatally, flawed. With the GCMS result now questionable, the labeled release result suddenly looked a lot more compelling. Its two positive results, the gas release and the sterilization control, were no longer contradicted by a definitive lack of organics. Now, this does not prove that Viking found life. It only proves that the main reason for dismissing it was probably wrong. The exotic chemistry explanation is still on the table, though it's never been successfully reproduced in a lab. So today, the Viking mystery has three competing explanations. 1. The labeled release experiment detected living Martian microbes. 2. The result was caused by strange, non-living soil chemistry. Or 3. Viking detected life, but another of its instruments accidentally destroyed the collaborating evidence. The original Viking data can't tell us which is true. The landers couldn't see cells, sequence DNA, or bring samples home. To solve it, we need to go back with instruments designed with the perchlorate discovery in mind. This brings us back to the ghost in the machine, the labeled release experiment. If the reason for dismissing its lifelike signal was based on a flawed, no organics result, what are we left with? The problem is, no proposed chemical reaction perfectly mimics everything Viking saw. Especially the way the signal was completely destroyed by heat, just like a fragile living system would be. This opens up a wilder possibility. What if the Viking results weren't just a hint of Earth-like microbes, but a glimpse of a biology that operates on entirely different, more fragile principles? This 50-year-old mystery now directly shapes the future of Mars exploration. Scientists are designing new missions with instruments specifically intended to avoid Viking's mistakes and re-examine its bizarre results. So the Viking lander didn't give us a clear yes or no. It left us with the ultimate scientific cliffhanger. A clue, a blunder, and a question mark buried in the red dust. So did NASA find life on Mars 50 years ago? The labeled release experiment gave a signal that was consistent with life, and its result was confirmed by a crucial sterilization control. That result was dismissed because another instrument found no organic molecules, the no bodies, no crime rule. But decades later, the discovery of perchlorates revealed that this instrument likely destroyed the very evidence it was looking for. So the final verdict is this. Viking did not find definitive proof of life, but it found something that, 50 years later, we still can't explain. The honest answer is not yes or no. It's we don't know, and that is far more exciting. Because the implication is staggering. If life started independently on two planets in one solar system, it would suggest that the universe isn't just empty space, but a cosmic petri dish, and that life is not a miracle, but a common outcome. And it leaves us with one last tantalizing question. What if humanity's first evidence of extraterrestrial life has been sitting in an old NASA dataset for 50 years? If you like digging into scientific cold cases and asking the big questions, you're in the right place. Hit subscribe and join us for more smart stuff with stupid drawings.